G-protein coupled receptor, GCPR. This receptor are present in the cell membrane. GPCR has three parts viz. Ligand binding domain, where the ligand binds, membrane spanning domain, which has 7 spanning, intracellular domain, which is also called as effector domain. On the effector domain, there are 3 subunits present, which are called trimoric G protein. These 3 subunits are alpha subunit, beta subunit, gamma subunit. There are 3 types of G protein signaling pathway. G is G protein stimulatory signaling pathway. G G protein inhibitory signaling pathway. GQ signaling pathway. Let's have a look on GS signaling pathway. Ligand such as epinephrine binds to GPCR. It is a first messenger. After binding of ligand, the ligand binding domain gets phosphorylated. Subsequently, the membrane spanning domain and effector domain gets phosphorylated. The phosphorylation of effector domain leads to its conformational change. Due to this, the trimoric J protein present on it gets phosphorylated by conversion of GDP into GTP. This phosphorylated trimoric G protein dissociate and form 2 complexes viz. GS beta gamma complex and GS alpha subunit complex. The GS beta gamma complex will go and activate some ion channels. After activation of ion channels, the GTP will be converted again back to GDP, resulting in inactive beta gamma complex. This inactive complex will go back to effector domain and bind to it. The phosphorylated GS alpha subunit complex will go and bind to an adenylyl cyclase enzyme, due to which the adenylyl cyclase enzyme gets phosphorylated. Then, GTPase accelerating protein, GAPs, such as RGS protein, will bind to the phosphorylated alpha subunit complex, which is already bound to adenylyl cyclase enzyme. RGS protein will hydrolyze the GTP and will convert it into GDP. As a result of this, the alpha subunit complex will become inactive and will get separated from adenylyl cyclase enzyme and will return back to the effector domain. Now, the phosphorylated adenylyl cyclase enzyme will convert the ATP into cyclic AMP. Cyclic AMP is the second messenger. In the cytoplasm, there is protein kinase A, that is pKa enzyme, which is made up of 2 regulatory subunits and 2 catalytic subunit. The cyclic AMP produced by adenylyl cyclase enzyme will be released from it and will go and bind to the regulatory subunit of pKa enzyme. As a result, the regulatory subunit will get phosphorylated and it will undergo a conformational change. This conformational change will phosphorylate the catalytic subunit of pKa enzyme. Due to this, the catalytic subunit will dissociate from the regulatory subunit. This catalytic subunit of pKa enzyme will enter the nucleus through nuclear pores and will bind to inactive CREB present in it. This binding will lead to phosphorylation of CREB. This phosphorylated CREB will then go and bind to cyclic AMP response element of the targeted gene. The phosphorylated CREB then recruits a transcription coactivator called CREB binding protein, CBP, which is responsible for stimulation of transcription of targeted gene. After the binding of CBP, the gene will undergo transcription process to produce required RNA. Like, share, and subscribe to my channel. Thanks for watching.